So welcome everybody to our, I don't know how many years of the CBR Research Day. I think it's the 7th or 8th one. And it's obviously grown. We started off, I think, with 6 students when I arrived in 2009. And now we have 30 students this year and a lot of attendees at this event. So it's been great. So I welcome all of you. From multiple disciplines, from multiple faculties this year. Now we have biomedical engineers in the building, and we have scientists from science, from medicine, from dentistry. What did I leave out? There's a few. Anyway, so welcome to all of you. First order of business, please make sure your phones are on silent. Okay, I see all the students open up their phones. Okay. Great. So first I want to make sure we thank all the sponsors, the Neiman Vickers Endowment Fund, the Canadian Blood Services, CSL Behring, Bayer, Pfizer, Grifols, the Neil Mackenzie Memorial Fund, Macro Pharma, Octapharma, New England Biolabs, and Alexion and Stego. They're the ones who've supported all of the summer students to allow them to do these, to spend the 4 months or 3.5 to 4 months in labs, and so that it doesn't come out of the coffers entirely of your investigators. So thanks to all of them. In addition, we owe huge thanks to Julie, who particularly ran the CBR Summer Studentship Program, Mira, who's the office manager, and I'm going to take this opportunity to welcome Stephanie Mack, who's going to fill in Julie's position, who's leaving at the end of this month to move to Vancouver Island. So when you get an opportunity, I'm sure they're out there very, very busy. You've seen them lots. Please do thank them personally. So the program is, as always, the student presentations. Now we're shrinking the student presentations to, you really have to be flying through these presentations and extraordinarily eloquent and fast too, because you're down to 2.5 minutes. When we started, we were luxuriously giving 4 minutes, but I got a lot of concerns about sitting here through the night with 30 presentations, so we have shrunk them, but I'm sure you can get the messages across beautifully. That will be followed by the annual Neil Mackenzie Mentorship Award, and I think Dieter is here or will be here to introduce that award. And then we have a very special guest, Farah Alabay, who's come up from Los Angeles to give a talk. She tried it once before, but I don't know, her rocket failed, and her Air Canada rocket failed, so now she's flying United, and we're pleased to have her give a talk, and we'll hear more about that. And then that will be followed by an open bar, the posters, and that will be ready. As soon as you walk out this room, I promise there will be drinks, pouring, so particularly for the students, they'll have first shot. And then there'll be posters and poster judging as well. And then a barbecue dinner and awards, and many, many thanks. Now, here are the student presenters. That's all of you in the first 2 rows here, not to be worried. Some instructions on how to do this. Okay, first, do not look back. Okay, unless you want a picture of your ear or your ear when you're looking up there, look forward. This is a newly renovated room. Actually, we're the first to use it, as a matter of fact. So the screen you can see everything there, and the words, in case you're using the prompter, there are words on my computer, and I've just got the button. So you really have to stand here. You can't, please don't wander too far afield, because they won't hear you as well. So stay in this vicinity. All right, the target is 2.5 minutes. The hook comes at 3 minutes. Okay? And you'll hear a sound, okay, my chicken. All of the students are scoring each other. Score yourselves. Please put your name at the top. We won't divulge who you are. It's just so we don't lose track of how many scores we've tallied, okay? So as you go through, the students are scoring themselves, okay? And that will constitute the best oral presentation. That's all. Any questions? No, that's great. Okay, so we're ready to go. So our first speaker. All right, Cameron. Just so you know, it'll just roll through for the first 15, okay? That's your button.
Okay, got it.
Okay.
Okay. Hi, everyone. I'm Cameron from the Kim Lab. Now, if you guys can take a quick second just to think about pregnancy and the concepts behind it, it's fairly intriguing, at least to me, in that somehow your body can keep you up and running for 9 months, while during that time it's dealing with a newborn baby that's maturing inside. Now, if we take a step further and look at the immune system in pregnancy, it gets even more complicated in that somehow your immune cells can protect your body from disease, while that newborn baby that's maturing inside is technically foreign in the eyes of those immune cells. Now, all of that is still a very active area of research, but in general, what's known about the immune system in pregnancy is that your immune cells play a role by establishing different inflammatory states that can help the various stages of pregnancy progress. So early pregnancy is pro-inflammatory, mid-pregnancy and fetal growth is anti-inflammatory, and childbirth at the very end is pro-inflammatory. So these different inflammatory states that I'm talking about are governed by the local cytokine secretions of your uterine cells. And what's generally believed is that any imbalance or irregularities in these cytokine secretions can contribute to the pathogenesis of disorders like preeclampsia or preterm birth. So based on that theory, we wanted to see if we could find an accurate way to measure local inflammatory and angiogenesis markers during pregnancy. And that's where the umbilical cord comes in. So the umbilical cord is used to transport nutrients and oxygen from the mother to the fetus. And because it involves both maternal and fetal blood components, we felt it would give us a good perspective of what exactly the fetus is getting exposed to. So with that being said, what I spent my time on this summer was on running these multiplex ELISAs on samples of umbilical cord tissue that were taken post-pregnancy. And then using these ELISA kits, we were able to simultaneously measure 9 markers of inflammation or angiogenesis. And then using these measurements, we went back and looked to see if there were any associations with the pregnancy outcomes. So first we looked at whether the inflammatory markers that we measured in these cord tissues were correlated with chorioamnionitis or intraamniotic infection. And we did this to sort of validate what we're looking at. And as expected, every inflammatory marker that we measured had a significant increase in the concentrations of their cytokines. And one of our other findings looks at babies that were born too early or preterm pregnancies and something called VEGF or vascular endothelial growth factor. So VEGF essentially stimulates the formation of new blood vessels. And this finding here shows that babies that are born early had a 9-fold decrease in VEGF. So essentially this is suggesting that the impairment of angiogenesis could play a role in inducing early labor. So altogether, our main finding here shows that these umbilical cord tissue samples can be used to measure these different inflammatory cytokines and angiogenesis markers. And any of our other findings are very, very preliminary results that can help point us in a direction when studying pregnancy disorders that are not yet well understood. Thank you for listening, and come visit my poster.
Hi, my name is Colleen Wong, and my presentation today is on iron overload in patients aged 60 years and older with acute myeloid leukemia and how that may impact adversely on their overall survival. So just some background. AML is a malignancy in which abnormal production of blasts by the bone marrow occurs, leading to cytopenias. And AML is rapidly fatal, especially in those older than 60 years of age. Analyses in other anemias suggest iron overload portends inferior clinical outcomes, but there are no studies examining iron overload in older AML patients. So therefore, we performed a retrospective analysis of patients with AML aged 60 years or older and examined their iron status. So we had 101 patients with adequate data, and the highest correlation between the ferritin, which is what we called our iron stores, and the bone marrow iron store was at a serum ferritin level of greater than 750 and a bone marrow iron score of at least 3. And therefore, we defined elevated iron stores, which is what we called ELFI, as the following 2 parameters. So some of our baseline characteristics included the demographics, clinical presentation, our ECOG performance status, comorbidities, predisposing conditions, blood counts, marroblast percent, cytogenetic analysis, and cause of death. Marrow dysplasia and AML treatment received were the only factors that significantly differed between the 2 groups, and these were their p-values. So factors significant for overall survival in a univariate analysis were multiple. And then the factors with the trend included alpha, and there's its p-value of 0.07. And due to the limited patient numbers, we then did a pairwise comparison. And so the significant factors from the pairwise comparisons were then included in a multivariate analysis, which is shown below. Alpha and ECOG performance status had a significant p-value, and Alpha's p-value of 0.046 had a hazard ratio for death of 2.5. So this graph shows the overall survival by treatment received, and the median overall survival for supportive care and low-dose chemotherapy was 1.5 months, 12.3 months for azacitidine, and 16.5 months for induction chemotherapy. This just showed the subgroup chemotherapy, induction chemotherapy, and again, its overall survival for those having elevated iron was 10.1 months and 18 months for not having elevated iron. Despite small numbers in the subgroup, it reached borderline significance. So in this analysis for patients age 60 years and older with AML, elevated iron appears to portend inferior overall survival. And to our knowledge, these are the first data examining clinical outcomes in elderly AML patients according to iron stores. Thank you.
Okay. Hello everyone, my name is David, and my presentation is on how polyphosphate could be a potential therapeutic for age-related macular degeneration. So age-related macular degeneration, or AMD for short, is the leading cause of blindness in the elderly. There are two types, but I'll be talking about a type called wet AMD, which is characterized by something called choroidal neovascularization. So choroidal neovascularization is the formation of new leaky vessels in the back of the eye. This allows for an influx of leukocytes causing further damage and degeneration in vision. So AMD is a complement-mediated disease, which means that a key contributing factor is the dysregulation of the complement system. And polyphosphate is a molecule with known anti-complement and procoagulant activity. So that's why we thought that polyphosphate could be a potential treatment for AMD. The way we went about testing this was through an in vivo mouse model. We induced neovascularization by lasering the back of the eye, causing the formation of a lesion. After lasering, we would inject our treatment, collect the eyes, and stain them for new vessel formation as well as complement deposition. As you can see with our treatment, poly-P, the lesion size is significantly smaller. One of the ways we thought that poly-P was exhibiting this protective effect was by decreasing the influx of leukocytes to the site of damage. When we tested this, we found that polyphosphate did indeed decrease the adhesion and transmigration of leukocytes to choroid endothelial cells. So endothelial cells activated with inflammatory cytokines treated with polyphosphate showed a decrease in adhesion of monocytes, and monocytes treated with polyphosphate showed a decrease in migration of monocytes through those cells. So in conclusion, we can say polyphosphate could be potentially useful in the treatment of wet AMD in the future. And then the mechanism by which it elicits its protective effect is by decreasing neovascularization, so decreasing new vessel formation, dampening complement, and decreasing the influx of leukocytes to the site of damage. Thanks.
Hi everyone, my name is Ari from the Hancock Lab, and today I'm going to show you a project that I did where I screened D enantiomeric peptides for anti-biofilm activity. So biofilms are a cluster of bacteria enclosed in a matrix that are incredibly resistant to antibiotics. 65% of human infections are caused by biofilms, but currently there are no clinically approved treatments for biofilm infections. Fortunately, a group of peptides called host defense peptides have shown anti-biofilm activity. An example is the host defense peptide 3002, which can significantly decrease the size of a MRSA biofilm. But these host defense peptides have a problem. They are in the naturally occurring L form, which can be easily degraded by enzymes. Therefore, we looked into enantiomeric peptides, or d-enantiomeric peptides, and retroinversal, or RI peptides, that are resistant to degradation. D peptides are mirror images of L peptides, and RI peptides are D peptides, but with the amino acid sequence reversed. We hypothesized that these D and RI peptides can retain the anti-biofilm activity of the host defense peptides. We began this project by first obtaining the samples D enantiomer peptides in a spot synthesized array. Then we determined the concentration of the array peptides and selected ones for screening. Next, we performed anti-biofilm and cytotoxicity or hemolysis screening on the peptides, where we looked at their biofilm inhibition activities and also their potential for therapeutic uses. The graphs on the bottom shows results for a couple of peptides that I've screened. The y-axis shows percent biofilm growth, x-axis shows concentration of peptides, and L peptides are in black and DNRI are in red and blue. So as you can see, DNRI peptides can have reduced activity, same activity, or even increased activity compared to L-form peptides. You can see more of my graphs on poster 16. So my conclusion is that D-enantiomer peptides can have differing anti-biofilm activity compared to L peptides. And in the future, we can obtain these peptides at higher purity and confirm this activity, and also determine the difference in the mechanism of action for the enantiomer peptides. Thank you.
All right. Good afternoon, everyone. I'm Farshad Hosseini, third-year medical student at UBC, and I work with the BC Hemophilia Program at St. Paul's Hospital with Dr. Shannon Jackson, and great to meet you guys all. As many of you may know, patients with hemophilia A and B undergo a lot of bleeding at the joints, and that leads to a lot of recurrent joint arthropathy and joint degeneration. And because of that, many of these patients have to undergo a lot of orthopedic surgeries, including joint replacements, mainly the hip, elbows, knees, and the ankles. And what I did in our research lab was we followed all BC hemophilia A and B patients that have undergone orthopedic surgeries since 2005. And we looked at the type of procedures that these patients underwent over the 13 years, the amount of factor VIII that these patients utilized over the 13 years, the surgical complications, and the length of stay at the hospital over time. And we looked at each of the results separately. So types of procedures, we see that there's a transition from knee surgeries to ankle surgeries over the 13 years. And initially and there's also a lot of elbow surgeries as well. In terms of factor VIII utilization, so less and less factor VIII is being used by our patients over time as part of their management. And as you can see in the graphs, they're divided into 2004 to 2009 time period, which is the area of pre-prophylaxis, which is a different treatment regimen that we use currently. The 2010 to 2012 era is the transition period, and the final era, 2012 to 2017, is the current treatment regimen which we use currently, which mainly involves prophylaxis to prevent joint bleeding. And we see that because of this change in treatment regimen, we're using less and less factor VIII over time. We also compared the median length of stay at the hospital to our national values, and we're seeing that our BC values are pretty similar to our national values, which means that thankfully we're not falling behind from the rest of Canada. Surgical complications were all within expected limits. We see early joint bleeds, delayed joint bleeds, infection, thankfully no issues with thrombosis or clot formation, and some wound healing and mechanical and just joint pain complications as well following surgeries. And thank you all so much. And if you need me, you can find a number 28.
Hi, my name is Haley, and I worked in the Hancock lab the past summer exploring the effects of IDR1002 peptide on LESB58-induced responses. So host defense peptides are naturally occurring peptides that have a range of immunomodulatory and antimicrobial effects. Unfortunately, they can be cytotoxic and expensive to synthesize. So we have innate defense regulators, such as IDR1002, which are optimized for increased activity and lower cytotoxicity. IDR1002 was previously shown to have a protective effect against Pseudomonas aeruginosa in an acute mouse lung infection model. Therefore, we want to find out whether IDR1002 and antibiotics are synergistic against Pseudomonas aeruginosa. We used LESB58, a chronic strain of Pseudomonas. Human bronchial epithelial cells were infected with LESB58 at an MOI of 4 and treated with IDR1002 before incubation for 18 hours. Supernatant was collected to determine cytotoxicity and to quantify pro-inflammatory cytokine IL-6 and chemokine IL-8. A protective effect against LESB is desired to reduce cell death and to mediate IL-8 and IL-6 production. As it turns out, IDR1002 has a dose-dependent decrease in cytotoxicity. At 100 micrograms per ml, the peptide lowers the bacterial-induced cytotoxicity back to control levels. Curious as to whether IDR1002 has a synergistic effect, we showed that in combination, IDR1002 and ceftazidime, a beta-lactam antibiotic, is able to bring down the LESB-induced cytotoxicity to control levels. We further investigated how the combination of IDR1002 and ceftazidime would be able to mediate host responses in the presence or absence of LESB infection. Interestingly, we find that the production of IL-6 is suppressed in the presence of LESB. However, in combination, IDR1002 and ceftazidime is able to bring up the production of pro-inflammatory cytokine. At the same time, IDR1002 is able to mediate and increase the concentrations of IL-8 and is able to maintain the production when ceftazidime is used in combination. In the face of the declining effectiveness of available antibiotics, IDR1002 proves to become a possible therapeutic option by mediating IL-8 and IL-6 production in Pseudomonas infection. Future study can involve measuring anti-inflammatory cytokines at longer time points or quantifying recruitment of neutrophils by IL-8. Thank you so much.
Hi, my name is Helena and I'm from the Brome Lab. I spent my summer expressing mutant variants of human cathepsin K that are responsible for the disease pycnodysostoses. So cathepsin K is a protein mainly expressed in osteoclasts and is responsible for collagen degradation. It forms a complex with CSA or chondroitin sulfate, and it positively regulates the collagenase activity of cathepsin K. An overactive HCAT-K activity results in diseases such as arthritis and osteoporosis. A rare genetic mutation results in a misfolded or truncated protein which is unable to degrade collagen. It results in the disease pycnodysostoses that is characterized with short stature, osteosclerosis, and atypical facial features, as you can see from the picture above. Some mutation results in a stable and active protein that retains their active site and still have a decline in collagenase activity. The aim of my project is to investigate a mutant, namely the I150T mutant, and comparing it to a previously characterized mutant called the Y98Z, and comparing their collagenolitic activity. The hypothesis is that the I150T mutant would have a similar collagenase activity as the Y98Z, which is still lower than the wild-type protein. First of all, I introduced the mutation into the PKF vector using inverse PCR. I then transformed the PCR products into E. coli to amplify the plasmid. The plasmid is then purified and then transformed into PKF for expression. The colonies that that manage to integrate the plasmid is then cultured in a flask, and the protein is harvested from a supernatant as it is secreted. So the ZFR MCA kinetics of the wild type is shown above, and it's a fluorescent substrate that allows us to measure enzyme kinetics. The results shown for the Michaelis-Menten kinetics and CSA titration gel is consistent with what our lab have previously obtained. As for the mutant, unfortunately, there wasn't enough yield to purify, and we predicted that it will have a lower ZFR MCA activity compared to the wild-type protein. In the future, using the fermenter or an alternative expression pathway such as E. coli would help us increase the yield and allow us to purify the protein. If we manage to obtain enough yield to purify the protein, and we can use these methods to characterize it and investigate deeper into how the mutations can affect the disease. Thank you.
Hello, my name is Joshua de Guzman, and I'm one of the co-op students of the Hancock Lab. So I'm pretty sure most of you are aware of tuberculosis infections in humans. So my project involves Mycobacterium bovis, which causes the disease bovine tuberculosis, a chronic disease that primarily affects livestock. This disease results in $3 billion in economic losses annually worldwide and $10 million in Canada every year. And due to the lack of effective vaccines, practices such as the overuse of antibiotics and the slaughtering of infected animals have become common. So the goal of the revamp project, which I'm working on, is to use a genomics-based approach of identifying proteins which have potential immunogenic properties, which will be cloned, expressed, and then purified, then sent out for screening in animal models, and ultimately used to create effective vaccines and diagnostic tools. So the first portion of my project involved developing or optimizing growth and protein expression conditions in E. coli. So generally growth conditions were, growth media had the greatest effect on growth expression conditions. So as you see in figure 1b, protein 6 had the highest TB, which is shown in figure 1b for protein 6. So media had the greatest effect on protein expression conditions, while other variables such as temperature and growth time had little effect. That being said, if you look in the table to the left, the addition of glucose and the use of a lower concentration of IPTG, which is used to induce expression, more than doubled optical density of the culture. So therefore, conditions that were optimized for both expression and culture growth were used for scale-up expression. And then after growing these cultures, they were lysed and then sonicated. And these proteins were purified using his-tag affinity binding to a nickel NTA agarose resin. So essentially, it consists of a C-terminal his-tag, which is 6 histidine residues, which binds to the nickel ion via coordinate bonds, and the nickel ion itself is bound to the NTA resin. So once bound, these proteins were eluted using a pH gradient, and as the his-tag loses its ability to bind at a lower pH. So as the pH lowers, it starts to get unbound. And ETA is added in the last few fractions to completely remove the nickel off the column, and therefore any remaining bound proteins. And fractions were run on an SDS pH shell to confirm where exactly the protein eluted. And the protein amount was quantified using a Bradford total protein assay. So as of now, 3 proteins have been purified and successfully expressed, and two of them have been sent out for screening, and additional proteins will be purified in the future. So if you want to learn more about this protein purification, come talk to me at poster number 20, and thank you.
Hi everyone, my name is Kaylin, and I'm from the lab with the name that accurately describes its members. You guessed it, the Divine Lab. And today I will be taking you through my project that is looking at protein biomarkers for identification of pore storing red cell concentrates.
So red cell concentrates are used in transfusion medicine in order to increase oxygen delivery throughout the body. Now, the red blood cells undergo many changes over storage, collectively known as the red blood cell storage lesion. Thus, they can only be stored for 42 days. Hemolysis, which is the lysing of the red cells, is a major quality control measurement, with the maximum allowable amount of hemolysis being 0.8% per unit. Now, hemolysis is actually used as an indicator for red cell quality. Thus, as storage time and hemolysis increases, red cell quality decreases. Previous research in the Divine Lab looked at healthy donors exhibiting repeated high hemolysis and actually identified 3 protein biomarkers that were found at significantly lower levels in the high hemolyzers compared to controlled donors. These proteins are catalase, peroxyredoxin 2, and pan-20S proteasome. So, for my project, we looked at a random sample of 12 red cell units and aimed to determine whether or not these biomarkers can be used as an indicator for hemolysis in normal red cell units. So, each red cell unit was sampled on day 1, 14, 28, and 42, and osmotic fragility, morphology scoring, microvesical counts, and hemolysis were all measured as in vitro markers of the storage lesion. Ghost cells were then prepared by removing all the hemoglobin, leaving only the red cell membrane behind. And Western blots were then done using these ghost cells and were probed with antibodies specific to the 3 protein biomarkers. So, here we can see that as storage time increases, hemolysis, the osmotic fragility, and the microvesical counts of the red cell concentrates increases, while the morphology scores decreases. And all these changes that occur during storage are actually indicative of a decrease in quality of the red cells. Here we are able to see the protein bands for each unit, and a graph that shows percent hemolysis against the protein band intensities. Now, although there was no statistically significant correlation found between the biomarkers and hemolysis levels, there is an indication that for catalase, protein levels are lower with higher levels of hemolysis. So, in order to determine a significant correlation, a larger sample size, as well as more variation in hemolysis, is needed for future repeats of this experiment. And if a correlation is found between these biomarkers and hemolysis levels, then a new tool could be implemented for donors to assess the quality of the red cells and predict how well they will store over storage. Thank you.
Hello everyone, my name is Henry Wong, and today I'll be sharing with you my work in Kazakadathu Lab researching polymer therapeutics for transfusional iron overload. So iron overload is a common consequence of blood transfusions due to the fact that humans do not have a natural iron excretion pathway. And because of this, iron is essential, but it can be toxic due to the fact that it causes for the production of highly reactive radicals. Now, the current FDA-approved treatment for this is using iron chelators such as desferoxamine, also termed DFO. However, it's characterized by high toxicity, short vascular residence time, and also side effects. So, we at Kazakadathu Lab created an alternative chelation system through the conjugation of DFO with a biocompatible molecule known as hyperbranched polyglycerol, also termed HPG. My work revolves around investigating the iron chelation properties of this DFO HPG molecule, but 2 biocompatible versions of this molecule. So this was the experimental outline. We had 4 different treatment groups, whereby 2 of these treatment groups were the biocompatible polymers, also termed GHBK and DMK. And essentially, there were 4 mice put into each of these treatments, whereby each mice underwent a 21-day treatment. On the first day, they were injected with iron for iron overload. And on days 8, 11, 14, and 17, they received their respective treatment injections. Finally, on day 21, the mice were sacrificed and their organs were collected. Urine and feces samples were also collected daily from days 8 to 21, and all of these samples were analyzed for their iron content using an instrument known as inductively coupled mass spectroscopy, or spectrometry. And what did we find? Well, I'm glad you asked. On the left, we see that for urine, the GHBK polymer showed more amounts of iron excreted, whereas for the DMK polymer, there were greater concentrations of iron excreted in the feces, as seen on the right. When we look into the organs, we see that for the DMK polymer, it was associated with decreased amounts of iron in the liver, whereas for both of these polymers, it showed decreased amounts of iron for both the pancreas and the spleen. And putting these results together, we sort of see that there are 2 different iron excretion pathways that exist. For the first polymer, GHBK, it excretes iron via the kidney and then going towards the urine, whereas for the DMK polymer, it excretes iron via the liver, which then travels from the bile and then towards the feces. So overall, both of these polymers do show effective iron excretion properties, and it supports its potential clinical utility. If you'd like to learn more about the poster or my project, please feel free to drop by. Thank you so much.
All right, good afternoon everyone. My name is Kristen. The summer project is a collaboration between McNagley from the BRC and Moan from the LSI. All right. So it is becoming clear that our microbiota can program our immune system, and the microbial products can help maintain healthy homeostasis. The disruption of the balance may be causing allergic diseases such as asthma. So mice treated with antibiotic vancomycin they lack the bacteria required to ferment short-chain fatty acid, and they are more susceptible to allergic diseases such as asthma. So to test whether short-chain fatty acid exposure alters the behavior of blood cells, researchers in my lab transplanted bone marrow from the 3 mice listed on the slides, control vancomycin plus short-chain fatty acid supplementations to the irradiated control mice. What they found is that the Th2 skewing, as measured by the elevated IgE level from the vancomycin-treated mice, are transferred via bone marrow transplant, and the short-chain fatty acid supplementations can eliminate the skewing. So then they test if the microbiome alters the epigenome of a transplantable hematopoietic compartment. They use ChIP-seq to analyze DNA acetylations in a subset of stem cells to analyze from the mice isolated from the mice that raised from the 3 conditions. They found unique regulatory states are imprinted in the absence of short-chain fatty acids, which are pro-inflammatory. So the question for me this summer is what the exact population within the bone marrow are capable of conferring atopic immune phenotype. Could it be the hematopoietic stem cells or the mature lineage cells? So I did qPCR on the 2 populations. One is total bone marrow, and the other one is lineage depleted stem cells. In conclusion, I find that the mature lineage cells are capable of conferring atopic immune phenotype. So in the future, I would love to perform a single-cell RNA-seq on the mice treated with the 3 conditions, and in order to see if we can get similar results as the ChIP-seq data. Thank you guys very much.
Hi everyone, I'm Lisa from the Chung Lab, and today I'll be talking about gellan gum, which is a polysaccharide that many of you have probably not heard about, but had probably drank before. So my project this summer was to characterize gellan gum for suspending cells in single-cell printing. So first of all, what is drop-on-demand printing? Imagine your inkjet printer at home dispensing cells instead of regular ink. And you can use it for single-cell genome sequencing as well as potentially in tissue engineering. However, there are challenges such as cell reflection, in which the cells are reflected upwards instead of downwards in the droplet, as well as material deposition in the inkjet nozzle. So as an alternative, we have been investigating gellan gum as a bio-ink instead. It is a polysaccharide made by bacteria, and it has been identified as a bio-ink for its ideal characteristics as a Bingham plastic, which means that under low shear, it behaves as a solid to suspend particles, but under high shear, it acts as a liquid to be printed. And fun fact, it's found in aloe vera gel to suspend the aloe gel. My objective for this project was to characterize gellan gum for suspending cells during printing. So what I did was I first verified that gellan gum does reduce cell sedimentation. And I did this by suspending the cells in PBS with or without 0.1% gellan gum and imaged at the bottom of the well plate. And as you can see with the gellan gum, the cells remain suspended over time. Next, what I wanted to do was measure sedimentation velocity of 15 micron diameter polystyrene beads as a proxy for mammalian cells. And I did this in a capillary tube over a period of time. And with increasing concentration of gel and gum, the sedimentation velocity decreases, with 0.05% gel and gum being sufficient to fully suspend the beads. And finally, I characterized the fibers using black calligraphy ink with bright field microscopy. And the mean fiber diameters for the different concentrations of gel and gum were about 10 microns each. So in conclusion, gel and gum can be used to suspend cells for inkjet printing. However, since it's a non-Newtonian fluid, that means that it does not form uniform droplets as a Newtonian fluid like water does. And instead, you can see on the right with gel and gum. So in the future, we will try to sonicate the fibers to shorten and thinnen them to increase the ease of printability. And so if you'd like to get a better taste of gel and gum, come stop by my poster, number 11. Thank you very much.
All right. Hello, everyone. My name is Maria Elizabeth, and today I would like to discuss with you my summer project titled Are Eyes More Than Just Windows to the Soul? Exploring the link between age-related macular degeneration and Alzheimer's. So what possible links could exist between a common form of dementia and an eye disease? Well, both degenerative conditions do affect elderly people, and there is an accumulation of immunogenic deposits, amyloid beta in Alzheimer's and drusen in AMD. Previous studies have suggested a link between these two conditions, and recently it was found that retinal cells are a major source of amyloid beta. So we hypothesized that the eyes of TG2576 mice, which is the animal model of Alzheimer's, would demonstrate characteristic features of AMD. We proposed a model in which a decrease in complement factor H, which is an inhibitor of the complement cascade often dysregulated in AMD, would cause an increase in inflammatory complement protein, which would increase angiogenesis and eventually lead to the accumulation of amyloid beta and drusen. Now, this model is a simplification, but it gives us a good framework to develop our experiment. So what we did is we took the eyes of mice and we looked at protein using Western blots and immunohistochemistry. We looked at 5 key markers and links between Alzheimer's and AMD. Today I'll be discussing the results of the first 4. So what we found is that there is an increase in amyloid precursor and amyloid protein in the TG retinas. We also found increased expression of CD105, a protein associated with neovessels and angiogenesis. With regards to a link to AMD, we found increased lipofuscin, a component in drusen, in the retinas. However, we expected a decrease in complement factor H and an increase in inflammatory complement protein, but we saw no change in either. So in conclusion, we found that the TG eyes did have similar characteristics to those seen in the brain, increased amyloid and increased CD105, and we found evidence of increased drusen in the TG eyes. However, the immunological link between Alzheimer's and AMD is still uncertain. If you'd like to hear me talk about this project in more excruciating detail, please come and find me at my poster. Thank you.
Hi everyone, my name is Nabil and I've been working on delivery methods for hemostatic powders at Castrop Lab. So just to give you a bit of a background on the work I did this summer, upper GI bleeding is bleeding from the mouth to the upper small intestine, and it affects around 170 per 100,000 people per year with up to a 30% fatality rate. So these bleeds are managed endoscopically using clamps and electrocautery. And a recent advancement has been in sprayable hemostatic powders, which require less operator skill and can be applied in hard-to-reach places and for larger bleeds. Now, these powders are far from perfect. The ones available on the market now act as a sort of bandage, but do not apply any therapeutics to the site. So this has been shown to cause around an 80%, up to an 80% rebleed rate after treatment, leading to increased OR time, higher risk of complications, and an increased need for blood products. So Castrop Lab has developed a new topical powder called Counterflow, which by reacting an organic acid with a carbonic salt carrying cargo, CO2 bubbles are generated, which release the cargo, propelling them in all directions, including deep within the wound site. So then, can we adapt this technology for managing GI bleeds? So we hypothesized that Counterflow can be sprayed through an endoscope-compatible device to stop upper GI bleeds in swine models. So to test this hypothesis, my project had 3 objectives. First was to design a device that will transport Counterflow to the site of the wound. Now, these spray devices exist, but as to not infringe on existing patents, we had to make a completely new one to test our powder. Next was to make Counterflow sprayable, changing components to make the powder more suited to internal bleeding. And finally, it was to test in a swine model to determine the efficacy of the powder. So we are currently working on securing some IP around the project, so please excuse the vagueness of my results if we have yet to go public with the information. So I can't say much, but what I can say is that we have finally made a spray device. It took us 18 prototypes to get there. In optimizing the powder, a new organic acid has been chosen, which lowers toxicity and improves the bulk flowability of the powder. And we have adjusted the particle size as well as the carbonate salt to acid ratio. So in conclusion, Counterflow can be sprayed endoscopically and delivered to wound sites, and future direction will be testing in a swine model this early September. Thank you very much.
Good afternoon, fellow summer students and CBR members. My name is Nancy from the Cote Lab, and my project this summer was to launch a preliminary study looking at the association between HIV therapeutic interruptions and mitochondrial DNA mutations. So first, a brief introduction. So HIV therapy in the form of drugs has forever changed the landscape of AIDS and saved countless lives. Now imagine that there are 2 people prescribed HIV therapy. Person A follows their prescription strictly and takes no breaks. Person B, however, may have taken 1 or more breaks, and this is fairly common because many doctors will advise someone to stop for their own health reasons because these drugs are actually very toxic. So then naturally we wondered, is there a consequence for person B, who is actually the majority of people? So this leads to our objective, which was to investigate the effect of interruptions during HIV therapy on mitochondrial DNA mutation. And so why mitochondrial DNA mutation specifically? This is because these drugs, while they do a good job of inhibiting HIV's reverse transcriptase, they also inhibit our own mitochondrial DNA polymerase. So for methods, we took altogether 164 patients who were HIV positive and negative, children and adults, and we basically sequenced their mitochondrial DNA. And we were looking specifically for 2 types of mutations. The first were de novo mutations, and second was heteroplasmy. So to illustrate the difference between these 2 types of mutations, I have Jack to help me. Basically, de novo mutations are the ones that we all know about that occur randomly and spontaneously, just like how Jack can randomly and spontaneously turn into Fire Baby. And heteroplasmy is essentially mutations in the mitochondria that have been clonally expanded, just like how Jack can duplicate and make many, many copies of himself. So for our results, the de novo mutations first, we found that the number of interruptions is not associated with it. However, it seems that as you increase the duration of the interruption, you actually get an increase in the de novo mutation frequency. And for heteroplasmy, we found that neither the number of interruptions nor the interruption duration are associated. So in conclusion, heteroplasmy, number of interruptions and interruption duration not associated, but the de novo mutations are actually associated with interruption duration. And you might wonder how these results may change when you consider other things going on in a person's life. So for my future directions, I analyzed this data, but taking into account other factors such as aging and smoking, and we actually get some interesting and significant results. And if you want to find out what they are, please come visit me at poster number 3. Thank you.
30-second stretch break here while I change the thing.
fine. No chickens, fox.
Oh, well, I was being gentle. You okay?
No chickens, fox. Yeah. Keep people stretching, right?
It was like,
Yeah. The food was good. This was good.
well, it's the only thing. If you found good food,
Yeah. The barbecue's at 7. It starts at 7.
no.
Oh, okay. I was like, that's a little late.
You won't stick around. All right, we're going to get started.
Good afternoon. I'm Nico Werschler. I'm a fourth-year University of Waterloo student, and I'm working in the Zanstra Bioengineering Laboratory this summer. Let's talk a little about early mammalian and human development. Why work towards understanding this? During early human development, the embryo journeys through morphogenesis, a series of events giving rise to structurally complex and functional tissues that are necessary for human life. Gastrulation is arguably the most important event of morphogenesis, as it yields primary germ layer formation as well as development of the basic body axes. Having the ability to define and capture these mechanisms may give rise to engineering processes that are scalable, letting us design complex tissues and organs for implementation in clinics. For ethical reasons, human gastrulation cannot be studied in vivo, but in the Zanstra laboratory, we have pioneered a technique allowing human pluripotent stem cell colonies, upon relevant stimulation, to self-organize similar to that of the embryo in vivo. As you can see, we have primary germ layer development on the bottom right, brachyury indicating mesoderm, SOX2 ectoderm, and CDX2 extraembryonic tissue formation. As cool as this is, the regulatory aspects of this are largely unclear. That is what led Dr. Himanshu Kaul, a postdoc fellow in the Zanstra Lab, and I, to develop and validate a computational paradigm that kind of shows what happens predicts what happens in the gastrulating process. This tool did nothing less than it was designed to do, as the model predicts BMP4 and Wnt expression, concentration profiles, which are the ligands that are necessary for loss of pluripotency and mesoderm development, which is the germ layer that gives rise to tissues such as the blood and heart. This result allowed us to further predict expression profiles of SOX2, which is ectoderm, OCT4, our pluripotency marker, which thus explain the observed spatial germ layer organization, inward relocalization, and likely appearance of cells yielding the mesoderm trait. At the Zanstra Lab this summer, I have helped move this platform to 3D, and we expect to work more with spheroids to recapitulate this process computationally and experimentally. And as famous developmental biologist Louis Wolpert once said, it is not birth, death, or marriage, but gastrulation, which is truly the most important time of your life. Thank you very much.
Hello everyone, my name is Niman. I'm from the Rossi Lab. So while we're on the topic of muscle regeneration, I would like to talk about basically to better understand muscle regeneration. We must first understand the different cell lines that are involved in this. Muscle progenitors are most important for muscle regeneration, but they have other players that help in this process with them. For example, after injury, inflammatory cells infiltrate into the tissue from the endothelial cells and regulate both FAPs and also clean out the necrotic debris that exists within the cells, within the tissue. So among these cell types, we have the FAPs, which are very important, which I will talk about later. So we can understand that there is a vast intercellular network in these muscle that is involved in muscle regeneration. But one particular one that we worked on was VEGF, vascular endothelial growth factor alpha, which is known to be an important factor for angiogenesis, but its role might go beyond it. So actually a study added VEGF-A into muscle progenitors in culture and saw growth, as you can see on the right side of the slide, in these muscle progenitors. So to do this, we use a PAX7 VEGF knockout and the PDGF receptor alpha knockout to knock out VGFAs in these muscle cell lines. And we also use Natexin damage, intramuscular injection into these cell types to kind of understand how the regeneration works. So as you can see in here, the FAPs and MPs are producing the highest amount of VGF after 3 days of damage. These data were obtained from sorted cells. So we decided to knock out FAPs and MPs, VGF in these MPs and FAPs. And as you can see, the panel in the middle shows the muscle progenitors where basically the muscle tissue where VGF was depleted. And as you can see, compared to the wild type and the FAPs, the phenotype is different. And there is the phenotypic characterization is associated with impaired muscle regeneration. Also, we looked at fibrosis. And as you can see, the panel in the middle, which again is the VGF depleted muscle progenitors, shows increased adipogenesis and fibrosis. Also, we did some quantification that actually yielded very interesting results. I would love to talk to you about more on poster number 32. Thank you.
All right, hi everyone. I'm Olivia and I spent my time in the overall lab, mostly hunched over a microscope looking at gum tissue. So why gum tissue? Well, we're looking at periodontitis, which is the inflammation of the gums and the supporting structures around the tooth. So this is when plaque builds up, leads to tartar deposits, which allow for colonization of periodontal bacteria. This leads to chronic local inflammation, which eventually leads to gum recession and bone destruction in the tooth and in the jaw. So one mediator of this inflammation is high mobility group box 1. This is a moonlighting protein, which means it has one intracellular function, which in this case is its regulation of chromatin, and it acts as a transcription factor. But then when it's secreted, extracellularly, it acts as a immune system activator and cytokine modulator. Also implicated in inflammation is MMPs, and my lab has previously found that MMP2, 7, and 12 cleave HMGB1 into fragments, which then go to modulate inflammation and are predicted to be antimicrobial. So to start looking at this proteolytic processing, my goal this summer was to visualize HMGB1 and MMPs in the gingival epithelium. So basically what I did was take the gingival epithelium from wisdom tooth extraction. So my goal was to look at one piece of tissue that was inflamed and one that was healthy and compare the two. So I just sliced them all out really thin, fixed them to slides, and then stained them with either HMGB1 and MMP7 or HMGB1 and MMP2 and observed them under a microscope. So what I saw was HMGB1, shown in pink here, can be seen in similar distribution in healthy tissue and inflamed tissue. So in the nucleus and around the cell membrane or extracellularly. MMP7 likewise could be seen in similar amounts in both inflamed and healthy tissue, as shown in green in figures A and C. MMP2, however, could only be seen in inflamed tissues in figure D. So in conclusion, well, aside from everything I said just now, the main take-home message is that in fact, HMGB1 does colocalize with both MMP7 and MMP2, suggesting proteolytic processing in the gingival epithelium. Thanks.
Okay, good afternoon everyone. My name is Ryan Karimi and I was a summer student in the Kim lab, and my summer project focused on looking at PF4 and how it interacts with the main cell type in your gums. So Olivia before me did a great job at explaining what periodontitis is. Essentially it's a bacterial colonization of your gingival tissues which leads to chronic inflammation. Not to scare anyone, but if you don't floss, that will happen. So we've been able to show in our lab that platelets are activated by a molecule that's expressed on the surface of these bacteria called LPS, and from there they can release a whole slew of platelet-derived chemokines like PF4. Now we hypothesized that PF4 binds somewhere on these cells and transduces a signal to the nucleus, which eventually mediates a pro-inflammatory response. And my summer project is essentially looking at that teal oval and arrow, figuring out how the signal is transduced through the cell.
So the first thing we did was screening optimization. We cultured human gingival fibroblasts on 6 well plates and treated them with PF4 in a time course. So here's some of the different pathways we looked at first. We looked at the ERK pathway, the JNK pathway, and the STAT pathway. And after many, many blots and sleepless nights, we found that NF-kappa B does exhibit a phosphorylation at 1 and 3 minutes throughout our time course. We also took some confocal images to sort of optimize our antibody concentrations and incubation times. So NF-kappa B, unfortunately, is quite a downstream protein, so we did still have to look at a more specific pathway and trying to find what leads to NF-kappa B. After a lot more screening, we found that the P38 MAP kinase unexpectedly is dephosphorylated throughout our time course, so it's constitutively phosphorylated at baseline, but then as the time course progresses seems to become less phosphorylated. And then the confocal images there, again, verify that we were able to tag phospho-P38. And lastly, we looked at inhibiting the receptor that we hypothesized the signal is transduced through the CXCR3 receptor. So by doing, the way we did that was by incubating cells in a potent inhibitor of the receptor, AMG 487. And we found that this does indeed change the phosphorylation patterns of both P65, NF-kappa B, and P38, which heavily implies that the signal is at least partially transduced through the CXCR3 receptor. So for more information on all of our optimization and stuff like that, come see me at poster number 13. Thank you.
So hi, my name is Ryan Solomon, and I was in the Kazakh lab this summer working on cryopreservations of human red blood cells using a novel polymer osmolyte. You may ask why cryopreservation? It's been around for the last 70 years, but the blood supply right now is at its all-time low. If you didn't know, as of August, we don't know if we're going to make it to Labor Day. So I was looking at new ways to try and freeze blood cells to keep them for longer periods of time because of shelf life. So I took whole blood and spun it down, just taking the PRBCs, and then with that I mixed it 1-to-1 with a polymer, and then cryopreserved that for a period of time, and then thawed that, and then did hemolysis to determine how well my cells were doing. HPG is the polymer that I was mostly using. There's a lot of proprietary stuff, so I can't talk about it too much, but it's hyperbranched polyglycerol. There's 3 main parameters that we looked for: molecular weight, polydispersity, and the degree of branching. I used many different kinds of molecular weights to see what the best fit was for the cryopreservation. And if you don't know, I probably took your blood at some point this summer. I did over 100 different assays, so if you know me, I'm sorry. So my results were actually pretty promising. The green shows the FDA-approved current cryoprotectants that are used today. The pink and blue and purple are the HPG molecules and HPO molecules that I used. I was able to get less hemolysis using the polymer for a period of 1 hour and then a period of 1 week, and then moving forward to a period of 3 weeks. So this shows promise in the use of certain polymers as cryoprotectants. Then I got more ambitious, and I was like, let's do a lower storage temperature. So I went to negative 80 degrees Celsius using dry ice and acetone, creating a slurry. And this showed promise with certain polymers, but not other ones. So HPO 1K was able to show equivalent lysis to glycerol, which is the most commercially used available cryoprotectant. And then I wanted to get an image of my cells to see what was going on with the actual crenation of the cells. So after a period of 1 hour, you can see the cells still have their concavity. They're not too bad. And this shows promise that if they do get transfused into a patient, they can be functional. Furthermore, this application could be used for organs or different kinds of blood cells. And moving forward, I'd like to get to negative 20 to see if we can keep that storage low for better transportation. It has great ability to increase the blood supply, which is in great need. Thank you.
Good afternoon, my name is Sam, and this summer I was working on simultaneous single-cell phenotyping and chemotaxis assay under the supervision of Dr. Hongshen Ma. Now, before we get into that, I need to give you a quick background. Chemotaxis is the idea that cells move in a chemoattractive gradient towards the source. This is important going through my slides. So the objectives going forward was to create a software that can simultaneously phenotype and track cells in a chemoattractant gradient. And we decided to do this for an upcoming clinical trial that we're working towards. Sorry. So the single cell tracking side of the software we take a series of images on a microscope and then we feed them into the software. These images are then passed to and where they are processed to determine the cell paths of each individual cell, as you see in the image up here. From these cell paths, we're able to extract data on their speed, direction, and the linearity of their motion. Once we've tracked the cells, we then pass it over to our phenotyping. So the tracked images are cross-referenced with the stack of 3 fluorescent images to determine their phenotype. An extra function was added to compensate for image misalignments due to post-chemotaxis in vitro staining, which is required for clinical experiments. All of the data is then saved to Excel for easy data analysis by the scientists. I should also note that this software was written from scratch using C# and consisted of some 2,000 lines of code. So to test the software, which was somewhat important, I took 3 separate groups of the same HL60 cell culture and stained them with blue, red, and green. The cells all originated from the same cell culture, so we expected to see similar velocity results. The more important result that we were looking for is that we were able to identify the separate phenotypes by their dye and differentiate them. So to conclude, the chemotaxing cells could be tracked using the software, so it works. The cells could be identified and separated based on their phenotype through fluorescent staining. And the future application that we're building towards is a clinical trial for determining the effectiveness of chemotherapy drugs on targeted tumor cells harvested from a mouse. Thank you. And for more information, see me at poster 15.
Hello everyone, my name is Sam as well, and I'm a summer student at Dr. Kelly McNagny's lab. We were interested in studying the role of IL-22 in the context of inflammatory bowel disease. IBD is a chronic disease that is characterized by severe inflammation, thickening of intestinal walls, and ulceration. It involves dysregulation of Th1 and Th17 responses in the gut, and IL-22 is one of the cytokines that is involved in these responses. It's produced by Rohr gamma T cells and acts exclusively on epithelial cells, especially in the barrier tissues. It's known to be critical for inducing antimicrobial peptides, formation of the mucus layer, and finally establishing the host bacterial homeostasis. However, its role in the chronic state is unclear, so we decided to investigate by suppressing IL-22 in the context of Crohn's disease. We hypothesized that IL-22 has a role in preserving gut epithelium and pathogen clearance during the chronic infection. As a model, mice were pre-treated with streptomycin prior to the oral infection, and by day 7, mice were injected intraperitoneally for 3 times a week with anti-IL-22 or anti-ragbit antibodies, and they were finally sacrificed at day 21 and day 42 for immunohistochemistry, histology, and cytokine profiling. As you can see in figure A, we've discovered that neutralizing IL-22 leads to a severe disruption of intestinal epithelial barrier compared to the isotype control in at day 21. Furthermore, it showed us that there's a depletion of epithelial precursors at the bottom of the crypts, and figure B shows us that despite the severe ablation, the crypts were able to restore themselves by day 42. We also prepped the cecal tissues for mass trichrome staining and ELISA assays. The histology showed us that when the IL-22 is suppressed, there is a severe stricturing phenotype, as you can see in figure A. And also the cytokine profiles show that there's an enhanced Th1 and Th17 responses, indicating a heightened inflammatory response during the infection. In conclusion, IBD is characterized by dysregulated type 1 and type 3 immune responses, and IL-22 has a role in mediating intestinal epithelial barriers and progenitors. Additionally, it also limits the magnitude of inflammation, as you saw in the slides before. And going forward, boosting IL-22 response while suppressing other cytokines that's involved in IBD may be a potential therapeutic target for patients. Thank you.
So good afternoon, everyone. My name is Sarah, and I've been working in the Brown Lab at BCCHRI. Deficiency of adenosine deaminase 2, or DATA2, presents in children and is associated with blood vessel inflammation and stroke, which can lead to severe disability and death. DATA2 does appear to respond to therapies that target specific molecules. However, it has a broad clinical phenotype, including patients previously diagnosed with vasculitides, autoinflammatory diseases, and immunodeficiencies, making diagnosis a challenge. Although it's caused by mutations in the gene encoding the adenosine deaminase 2, or ADA2 enzyme, targeted DNA sequencing is not currently an optimal diagnostic tool, as not all pathogenic variants have been identified. The disease progresses rapidly, and recently there was a patient that passed away while waiting for sequencing results. There is a commercially available assay that can be used to demonstrate a loss of ADA enzyme activity in plasma and serum. However, it's not specific for ADA2 activity and was optimized for use with HPLC, liquid chromatography, a method that's not accessible at all hospitals. Also, it does not allow for the interpretation of individual cases due to lack of standardization. So you imagine you get your blood test results back and you don't have a range to compare your glucose or your iron levels to. The results are meaningless. ADA2 activity has been shown to decrease with age, so the establishment of age-appropriate reference ranges will be critical. The assay is based on the enzymatic deamination of adenosine followed by a series of reactions leading to the formation of a dye. So the greater the change in absorbance, the higher the adenosine deaminase activity, or ADA. Through a number of optimization experiments, we have established a protocol for a simple 96-well plate. Our optimized assay is really great in a lot of ways, so please do come ask me more. As an example, one main downside of the commercial assay was that it was not specific to ADA2 activity. So we have ADA1, it's an intracellular enzyme, but with an 1,000 times higher affinity for adenosine than ADA2. Using EHNA, an ADA1 specific inhibitor, we can look at ADA2 activity specifically. We've shown that even in samples in which we induced hemolysis, where the activity is much higher due to the released ADA1, EHNA still inhibits the activity to the same level as seen in our normally processed sample. With our optimized assay, we were really excited to be able to run 46 pediatric control samples provided by the BCCH Biobank. We saw a clear trend towards lower activity after 8 years of age, with increasing age correlating to decreasing ADA2 activity. In summary, we have a robust activity assay, but due to the large variation within groups, we would not be able to use these values as reference ranges. We are now developing an assay to quantify the ADA2 protein of samples using immunoprecipitation followed by Western blot. Being able to normalize ADA2 enzyme activity to total ADA2 protein may help us tighten up our groups and answer the broader question of why ADA2 activity appears to decrease with age. Thank you.
Hi everyone, I'm Sarah from the Divine Lab, and my project is Analyses of the mTOR-regulated protein synthesis in human platelets. At this point, little is known about the role of protein synthesis on platelet in vitro quality. Though platelets are a nucleate, they are able to translate mRNA derived from megakaryocytes. A previous study in our lab demonstrated that protein levels increased from day 1 to 7 of platelet storage, but the mechanism for this was not investigated. The pathway of interest in this study is the mammalian target of rapamycin, or mTOR pathway, which controls protein synthesis. Rapamycin is an inhibitor that blocks activation-dependent phosphorylation of mTOR pathway components, and we use this to assess the downstream effects on protein expression levels and platelet quality. Aliquots were taken from Buffy-coat platelets on day 1 and 2 after collection. We created an in vitro model using agonists to activate these platelets. Samples were treated with low amounts of pyrimycin, which is incorporated into the nascent protein upon translation in order to monitor synthesis, in the presence or absence of rapamycin, or left unspiked as control. And from there, I ran these samples on Western blots. Additional tests, including flow cytometry and aggregometry, were done to look at the effects of rapamycin on protein expression levels and platelet quality, respectively. So the gel on the left shows that pyrimycin is well incorporated into nascent proteins after thrombin activation, as seen in the first lane. In the final lane, spiked with rapamycin, we see reduced pyrimycin incorporation, which is a measure of reduced protein synthesis. On the right are gels that show that TRAP6 activated platelets have an increase in protein expression, while rapamycin subsequently reduces this level. So what are the implications of reduced protein levels in these activated platelets? Flow cytometry to measure activation of platelets after TRAP6 activation showed rapid degranulation as measured by C62P expression. This effect was diminished upon rapamycin spiking as indicated by the red arrow. We also performed aggregometry by adding ADP, allowing us to obtain a dynamic measure of platelet aggregation ability. Compared to the red control line, rapamycin showed a concentration-dependent reduction in aggregation ability, from 55% to approximately 3%. Ultimately, our Western blot results show that rapamycin treatment reduces puromycin incorporation, indicating reduced protein synthesis. We also saw a reduction in specific proteins. Protein synthesis in platelets also appears to be linked to platelet functions such as degranulation and aggregation. There are applications of these findings to platelet blood bank storage to assess whether protein synthesis capacity affects in vitro quality and subsequent transfusion outcome. There are also further implications of this to reducing the platelet storage lesion. Thank you.
Hi everyone, my name is Sunny and I worked in the Conway lab this summer finding a new treatment for AMD. AMD, age-related macular degeneration, as a couple presenters have already talked about, is a disease in the eye. You can see from the picture how it affects vision. It is the leading cause of blindness in the elderly and affects 1 million Canadians. And with the aging population, that number is expected to increase. There are two types of AMD, wet and dry. For the dry form, there is currently no treatment, and for the wet form, the treatment is only 70% effective. AMD is a complement-mediated disease, which means that the complement dysregulation will contribute to the pathogenesis of AMD. Now, complement are a set of plasma proteins that defend against foreign pathogens. There are 3 pathways that activate the complement system, but they all involve a series of protein cleavage events that lead to the terminal pathway and result in cell lysis. Now, my goal for the summer was to find safe anti-complement molecules that could potentially treat AMD. The class of molecules I looked at are heparins. Heparins are polysaccharide molecules of varying chain length. They're made up of sulfated disaccharide monomer units, and they're most well known for their anticoagulant activity. I worked with these 6 heparin polymers and did a variety of different assays. The first assay I did was the hemolytic assay, which measures the complement activation in the terminal pathway. The amount of red blood cell lysis is correlated with complement activation. In the graph, the x-axis is the concentrations of the different polymers I used, and in the y-axis is the percent cell lysis. As you can see, the 4 polymers highlighted in blue are able to decrease the amount of cell lysis at very high concentrations, indicating that they have anti-complement activity. Of the 4 polymers, 2 were identified as safe, and I did the C1 inhibitor assay, which measures the amount of C2 cleavage in a purified system. Now, the cleavage of C2 is important for complement activation. On the gels, you're able to see with increasing concentrations of the 8-mer5 and the 12-mer5, which are the 2 heparin polymers, the cleavage product C2 band disappears. This indicates that my 2 polymers have anti-complement activity. In conclusion, I was able to screen 6 heparin polymers and identified 2 with anti-complement activity that acted through the terminal pathway and through C1 inhibitor. Now we want to use these 2 polymers in wet AMD mouse models to see if they are also able to work in vivo. And if you have any more questions or would like to hear about this in more detail, you can come visit me at poster 4. Thank you.
Hello everyone, my name is Trevor Yu, and this summer I worked in Dr. Karen Chung's lab, and my project was looking at polylactic co-glycolic acid for applications in an airway-on-a-chip device. So first, a little bit of background on airway-on-a-chip. We are designing an airway-on-a-chip model as an improved model for COPD. As COPD currently has no known cure, but current models are unable to model the complexity of the lung tissue in the small airway. That being said, our model does require a biodegradable membrane in order to make the tissue structure. So polylactic co-glycolic acid is a biodegradable polymer, and it's easy to form into membranes. However, in previous applications, it has been found to degrade over weeks to months. Ideally, for our applications, we'd like the polymer to degrade within 7 days, and that was one of the studies we conducted, which characterized the degradation. And we also wanted to ensure that our polymer would be suitable for cell culture and wouldn't have any negative effects on the cell lines that we used. During our degradation study, we filled our area-on-a-chip device with media and had our polylactic co-glycolic acid membranes in the chip, and we looked at the degradation under phase contrast images. We found that the membranes experience heterogeneous degradation with the formation of pores, wrinkling, and area loss as seen as an example. We also looked at the outlet pH of the media, and we found that the pH decreased when we had chips filled with media but had no perfusion. During our biocompatibility study, we found that our cells had the same cell viability compared to control. However, there were lower cell counts in the samples that had PLGA in them. From this, this indicates that for in-chip studies, we should seed cells at higher concentrations or allow them to grow longer to reach confluency. But overall, PLGA is suitable for our application as an in-chip separation membrane. In the future, we would like to characterize the degradation by having our chip loaded with hydrogel in the bottom channel, as this is more relevant to our model, and eventually construct a partial area on a chip co-culture with our 2 types of cells that we used. If you'd like to learn more about our project, please come visit my poster number 13. Thank you.
Hi, my name is Victoria, and I'm from the Hancock Lab. I've been working on the REVAMP project and focusing on expression and purification of Mycobacterium bovis potential antigenic proteins. So the REVAMP project is a reverse vaccinology approach to combating bovine tuberculosis. This disease is caused by Mycobacterium bovis and affects the lungs of cattle. The economic impact is severe, and currently the only method of prevention is destruction. For example, most recently there was an outbreak in Alberta costing $39 million and the preventative destruction of 11,500 animals. A solution? Vaccine that would provide cost-effective prevention. And this project employs a reverse vaccinology approach for vaccine development, first screening the genome of M. bovis to predict all potential protein antigens, then purifying those antigenic candidates for further testing. My job is to purify the M. bovis candidates. However, many of these proteins don't readily overexpress in E. coli, so I've also been exploring the use of maltose binding protein as an expression enhancer. Inducible expression tests were conducted first to obtain overexpression of candidates, and which should look like figure 1. And if successful, those candidates would then be purified by nickel affinity chromatography via a His tag. Purified proteins of sufficient concentration and purity would then be sent to Vito Intervac for further testing. However, a lot of my proteins did not do that because on figure 2 you can see a candidate that had co-purifying contaminating bands, so these proteins do not qualify. And for proteins that do not readily overexpress, I cloned them into dual MEP and His tagged gateway vectors. And for that, I focused on expressing 2 different candidates as fusion proteins to MBP. But when I conducted the same expression tests, the only overexpression bands I see on my gel and my Western blot occur around the same size as MBP, at around 43 kilodaltons, rather than the expected fusion protein sizes, such as 89 kilodaltons on this figure. And from this, it appears that only MBP is being expressed, which led me to believe that my gateway reactions failed and there's no gene insert in the clone. However, the gene was confirmed present when I ran a PCR, so I am not sure why I'm getting this kind of result. So far, it's been very problematic working with these MVP gateway clones and trying to express the fusion proteins. So as of now, the effect of MVP on the expression of M. bovis candidates is inconclusive. The next steps are to continue troubleshooting those results, perhaps try another expression host or work with other tags such as the leukotoxin tag that we just received from USASC. And the ultimate goal is to find a fusion tag that will result in the expression and purification of M. bovis candidates in high yield. Thanks, y'all. Please check out my poster.
Good afternoon, everyone. My name is Zach. I work at the Hancock Lab, and my project is investigating the antibiotic susceptibility mechanisms in Pseudomonas aeruginosa. So a little bit of an introduction, and I'm sure you, your colleagues, and your cat realize that antibiotic resistance is a very prevalent issue and evolves very quickly. One of the MDR pathogens that I worked with is Pseudomonas aeruginosa, an opportunistic gram-negative pathogen that forms potentially lethal biofilms in the blood, the skin, and biomedical devices. I also perform most of my tests in host-mimicking conditions because synthetic lab media such as MHB is often not representative of the in vivo environment. So to study the genes of the resistome of Pseudomonas aeruginosa, the bacteria were grown in host-mimicking media with azithromycin, and their RNA was harvested and compared to untreated samples. So these systems that we identified was the FOP and FOQ complement two-component system, which regulates the on operon. Those are highlighted in blue. And these systems are responsible for LPS modification that decrease the cell-promoted uptake of cationic peptide antimicrobials. And in pink, you can see these are drug efflux proteins, MEX-X and MEX-Y. These are hypothesized to facilitate the efflux of azithromycin out of the cytoplasm. So in order to study these resistance mechanisms, I created knockout mutants by clean deletion. And these, I also created some complemented strains and overexpression strains for the purpose of phenotypically comparing them to Pseudomonas wild type to assess their antibiotic susceptibility. A preliminary MIC assay, so an MIC assay is the minimum inhibitory concentration, so it's the level, it's the concentration of azithromycin that you do not observe any bacterial growth. So what I did was I used, with these 4 systems that I found, I performed a separate MIC assay in different types of media, focusing on host mimicking media. And you can see in the red boxes that for the knockout mutants, there is a greater susceptibility to azithromycin in host mimicking conditions. And next, I'll be focusing on synergy assays between antimicrobial peptides such as DJK5 and DJK6, and I'll be investigating their synergy with azithromycin, which could lead to increased potential for therapeutic treatment against Pseudomonas infection. And if you'd like to learn more about my work, don't come to my poster because I don't have one, but I'd love if you come talk to me anyway.
So Duke is asked, which brings us to, I'm not going to give Duke's talk. Kelly, you want to give Duke's talk? Okay, Kelly. No guts, Kelly. You couldn't do it in 2.5 minutes, probably. All right, we'll move it forward. It's spectacular, though, I'm sure. Okay. So I'd like to, I think we can all thank all these great students for a terrific job. And now that you've done this, you recognize that this was webcast and probably picked up in Russia and China by all our friends in those places. So I'd ask all the students now to complete the scoring sheets. Please do the addition at the end so that Julie and Mira don't have to do that, and then just hang on to those or just leave them on your desk, okay? And then we'll continue on. All right, so that was terrific. It's really remarkable. In 2.5 minutes, they can explain everything they did in 3.5 months, but it just points to eloquency and it points out to all the terrific science that's going on. Just take a breather. Okay, I don't want anybody to leave yet. Lock the doors. So the next piece on our program is a recognition of Neil Mackenzie, and it's a Neil Mackenzie Mentorship Award. But just to describe or tell a little story about Neil, I'm going to hand this over to Dieter Brummer, who's an investigator at the CBR, who was Neil's mentor and supervisor.
So yeah, it's time again this year to provide the awardee for the Neil Mackenzie Mentorship. So Neil was in my lab a postdoc. He joined around in summer of 2013. And so he came from Scotland, a typical, actually British or Scottish guy. So he was really great in the lab. He was very effective, brought a lot of ideas with him. We got even a CIDR grant for it. But he couldn't really complete his work. So he was a great guy in the lab, and he was, I think, even a greater guy in the outdoors. He was every weekend in the mountains. And he came back frequently with, some blue eyes and some, bendings in the arms and this kind of things. And yeah, unfortunately he overdid it at some point, and he crashed with two friends down in Joffrey Peak. I don't know if you know the area, and all of three passed away. So that was really a major loss for all of us. And since Neil was really also a great mentor, not just postdoc driven scientist, and so we thought about to keep his memory alive and provide you a mentorship award. And the mentorship award is provided really by you. So you elected your best mentor. It's normally a graduate student or a postdoc. And we keep this tradition, and today's winner is...
Oh, hold on a second. So I'll just take over for a second. So there were many elected nominees for this award. I hope I included everybody, or if I didn't, nab me after. But and it was clearly a difficult decision. This is adjudicated then by outside people who knew Neil, so are not necessarily, they were really from the Vancouver climbing establishment, who knew Neil in a different way than probably many of us did. So, and just to give you an idea of some of, I like to sort of list some of the comments about what these mentors were like, and you can see some of the descriptions: supportive, encouraging. I won't go through them all. Never doubts my ability. These are from you describing these mentors. Never always complimenting, nurturing, caring, empowering, considerate, caring, always there to talk science or life, eager to teach, and generous, open-minded. I always like that last one: pushes me to step out of my comfort zone, but always a half a step behind to provide support. So you can sort of look upon this, the mentors out there, and you who will be mentors should take a look at these sort of comments and on what makes for a good mentor. There's many, many components. So this year, the awardee, and these are some of the comments made, no matter the time of day, he makes time to discuss the issue that is holding your science back, giving helpful hints and suggestions, or maybe just to listen to the issues of your day. His passion for science is always evident, a wonderful person to talk to and get to know. And when you don't know the answer to the question, ask Fred. He'll have an idea or two, and will also suggest other people who would be able to help. An amazing mentor. So Fred Roselle.
Thank you very much.
Do you have anything to say to all these mentees?
I've been here for a long time, amazingly long enough to know Neil. He was a great person. Didn't speak much with him outside of the lab, but what I saw from him in the lab, it's a high standard. He set a high standard for all of the nominated mentors to live up to. There were a lot of cool people in that list, also Scott, all of whom deserve this, and even more people. It's actually very touching. So what makes one person the winner for this honor? And I think it boils down to the student who takes the time to listen and to seek the advice, and then to write the nomination. Whoever wrote my nomination, you've got a career in grant writing, I think. Thank you very much.
I for sure did not write anything, but I can agree that Fred is a really great guy. He was helping a lot of my students to get around in the tissue culture, or particularly in the fermenter unit. So he is really the best awardee this year.
Thank you.
Kitty wants to. Very good. Okay, so just let me load up the next slide deck. Take a breather, everybody. Hold on. Okay, so we'll get going again. Okay, all the students have had their little fix of their phones, okay? So now you can close them again. I know. It's hard to keep the hands off. Okay, so if there was ever a contest and you had to ask my wife what movie do I like the most, what's my favorite movie of all time, and undoubtedly she would say, what would you say, Ruth? Contact, exactly. There you go, you see? Okay. So that's, this is a story, based on Carl Sagan's book about, and then represented in a movie called Contact. The story of a little girl who, through her father primarily, becomes enchanted with space. And Farah's story is actually very similar, but this is actually for real and much better. So Farah grew up in Montreal, and then the daughter of immigrant parents from Madagascar, who obviously her parents obviously valued education, teaching, and the concept of giving back. And she was 8 years old, as she describes in one of her blogs, when Apollo 13 took off, but obviously it had a huge impact. She went to high school in Manchester, England, and then studied aerospace and aerothermal engineering at Trinity College at Cambridge University in the UK, completed her bachelor and master's degrees there, winning numerous awards along the way and obtaining truly the highest grades achievable at the university, which is no small feat. During that time, she took 1 year and spent it at Boston's MIT, well known obviously, where she developed several projects, but one of which was payload hardware for MIT's Mars Gravity biosatellite. And I'm sure she can fill in gaps in this little story because I have no idea what this means. In 2010, she entered into her PhD studies in space systems engineering at MIT. Her thesis being on, and I'll read this, on the evaluation of multi-vehicle systems for exploration of planetary bodies in the solar system. So it sounds like she was just, creating dune buggies for other planets. Her projects included the development of tools for multi-robot planetary surface exploration and technologies for a reusable lander. Since 2014, Farah has been a systems engineer for the Jet Propulsion Lab at NASA. Now, I didn't realize that NASA has many sort of locations throughout the United States. I always thought it was either Cape Canaveral or Houston, which is where it took off and landed. But there's a lot more to NASA than I know. She calls herself a jack of all trades, and it sounds like she really is. Her main tasks there, though, are to assess and help implement proposals for future NASA missions, space missions primarily, from the conceptual and design stages through to operations, resource utilization, and payload engineering. Now, recently she was the payload systems engineer for the InSight Mars lander, responsible for the instrumentation, providing test, launch, and operations support. This robotic lander launched in May of this year and is due to land on Mars November the 26th, I'm told. I'm not sure what time of this year, so watch for it. In one of her NASA blogs, she writes of one of her favorite projects, the solar mission to Saturn, which aims to look for life on Saturn's moons. So a real-life contact mission. FARA has received many awards. I only show 3 here, those that you might, at least one of them you might be familiar with, the Amelia Earhart Fellowship for Women in Aerospace Engineering, the Voyager Award for Marco Project Systems Engineering, and the Charles Alachi, I'm not sure I'm pronouncing that correctly, Award for Exceptional Contributions to the Lab. Charles was apparently the director of the lab for many years, led over 20 missions into space, but also is involved in Earth exploration, it seems. So he's quite the guy, and obviously quite an honor for FARA to receive this. She participates in all sorts of outreach activities, including mentoring students and teaching at space camps. She loves the outdoors and is an avid hockey fan. And I'm sure that she's either an LA fan or a Montreal Canadiens fan. She's probably certainly not a Leafs fan, undoubtedly, and probably not a Canucks fan, but nonetheless, we invited her here. So I'm welcoming Farah.
Thank you for the introduction. I am a Montreal Canadiens fan, so I mean we haven't won since I was a kid, so no one can really be mad at me, I guess. But so thanks for the intro. So yeah, I think I'm going to give you a little bit more of my background, and then today I actually I'm probably going to focus on talking about the InSight mission, which is the Mars lander that I'm currently working on. But I'll be around, so if you feel like asking me about any other mission you might have heard of, I will try to answer your questions. So I was born in Montreal, and I grew up just outside of Montreal. So that's actually a picture of me when I was 3. I grew up figure skating. I feel like every Canadian either is a figure skater or a hockey player, at least in Canada. In Montreal, that's what we do. There's not much else to do in the winter. It's cold. So I grew up there, actually only spoke French, and moved to England when I was 12 because of my dad's job. And that's how I learned English. So you can imagine as a 12 or 13-year-old, my parents would just say, go to school, learn English now. It kind of worked out. I guess it's a good way to learn a language. And I stayed there and then did my undergraduate and master's at Cambridge. Now, I was doing aerospace engineering at Cambridge, and the reason why I ended up in that job is because I watched the movie Apollo 13 when I was a kid. And I just remember my brother and I watched it together. My brother was a space nut. I really wasn't. I liked dinosaurs because there was another movie called Jurassic Park that was out at the time. Apparently movies just played a big part of my life. But we watched Apollo 13, and I saw these engineers, if you've seen the movie, right, they work together. And there's this big scene where they take all the parts from the spacecraft, put them out on the table, and try and put them back together to save the astronauts. And I came out of that movie and thought, well, first off, I want to be an astronaut because that's super cool. And also, I just loved the way that the engineers worked together. And I really, really liked the idea of working in groups and solving hard problems. And that's what led me to aerospace engineering. At the time, when I was about the same age, there was also a Canadian astronaut called Julie Payette, who you now know because she's pretty high up in the government too. So she was a huge inspiration for me as well, because one thing that was hard for me as a little girl from a small town in Canada is the concept of thinking that I could even work at NASA, right? How do you go from this town of about 100,000 people all the way out to working in LA? That's not a thing that I'd ever heard of, but I saw her do it. And so that really brought the concept of mentorship to me, and that's something that I practice today. She's really the reason why I do some of this, because I realized that she was such an influence to me. So I ended up studying aerospace engineering and decided that I really wanted to work more on the space side. And Cambridge was really focused on everything propulsion. So I ended up coming over the pond to the other Cambridge and moved to MIT, where I did my PhD. And I had the privilege to actually do quite a bit of hands-on research while I was at MIT. I worked with Draper Labs. Doc Draper, if you're not familiar with him, was one of the people that developed the guidance, navigation, and control for the Apollo system. And so he created that lab. I was actually taught astrodynamics by the guy that taught Neil and Buzz astrodynamics when they did Apollo 11. I actually met Buzz multiple times and got to meet Neil once even. So really developed my love for space there. And during that time, I also started working at JPL over the summer. So I was able to do part of my thesis out there and really applying it to real missions. So I fell in love with the place, as I feel like everyone does when they come to visit, and I never wanted to leave. So as an advice to you, if there's something you really want, go for it. There's no reason not to. You're going to regret it later. So I literally knocked on every single hiring manager's door that could possibly hire me. Now, I'm Canadian. Working at NASA was not really something you're allowed to do because I was a foreigner, but they do hire a few, as evidenced by me. If you nag them enough, they will let you in. So I went knocking, I made my case, and eventually, a couple of those people took a chance on me, and I ended up at JPL. So all to say that don't give up when you want something. There's really no reason to not go for it. So a little bit of background about JPL. A lot of people don't know that we have a NASA center in LA. So JPL stands for Jet Propulsion Laboratory. It is an FFRDC, so it's a federally funded research and development center. That means that I am actually an employee of Caltech. So I am a university employee and I'm a government contractor, which is one of the reasons why I could be hired as a foreigner. So I'm not a civil servant. And so JPL is a NASA center. The buildings are government buildings, but it is managed by Caltech for NASA. So that gives us some privilege in terms of what we're allowed to do and who we're allowed to employ, which makes it a very special place. Now, the name is Jet Propulsion Laboratory. We don't really build rockets anymore, but what JPL did do is they launched the first American rocket and the first American spacecraft, Explorer, into space. So they're the ones that built and launched that rocket. After the first sort of 10 years of rocketry in the US, so that picture down here is a picture of Explorer and Von Karman. And so after the first 10 years of rocketry, that part of aerospace work actually moved and was taken over by the Air Force and by other NASA centers. JPL became a NASA center, but its focus was to land things on other planets. So we landed the first moon landers in preparation for Apollo. We are actually the only institution in the world that have successfully landed on Mars, and softly on Mars, not crashed, which we've done too, but a lot of people have done that too. But some of the cool projects that you might have heard of, you've probably heard of Curiosity. That's our minivan-sized rover that we landed on Mars 6 years ago now. Cassini, that just ended a few months ago, was our mission that was around Saturn. We also have a mission called Dawn that uses electric propulsion at Ceres, and the Voyager spacecraft, which if you're Star Trek fans, you've probably heard of too. So we have two of those that are still in operation. Both of them are outside the solar system, and one is outside of the ecliptic. So I had the privilege to work on a few different missions, all the way from proposal stage all the way to delivery, and InSight has been the most recent one. So this is an actual picture from the InSight launch, which was on the 5th of May, so just a few months ago. This week is actually the halfway point. We've just reached a point where we are closer to Mars than we are to Earth, which is terrifying because that means we're landing soon. But this is a really cool picture, not the one I actually got to see. I was at Vandenberg, which is in California, right at the launch site about a mile away, and I saw nothing because it was foggy, which you're familiar with here, but still a really cool experience. It was the first interplanetary launch out of California. It's the first time we launched out of over the Pacific rather than the Atlantic in Florida. And there's reasons for that. You can come ask me why. I won't get into too much detail since there's a few more things I want to cover. Okay, but let's take a step back and talk about what InSight is actually going to do. So InSight is going to study the interior of Mars. So you're familiar with the Earth, we live on it, and you're familiar with the fact that the Earth has a core, right? It has a solid core, a liquid core, a mantle, and then a crust. Fun party fact for you, I don't know if you go to the same parties as I do though, but the thickness of the Earth's crust is about the same thickness as a tomato skin. Right, so that's how thin it is, and we know that. We know a lot about the Earth. We know it has plate tectonics. It's a very active planet. It has a magnetic field, right? We also actually know a lot about the moon because we've been to it. The moon's pretty dead. It's really boring. There's nothing going on. Has no activity. It's just kind of a piece of rock. Mars is somewhere in between. And other rocky planets are also different. Mars, as it turns out, has the biggest volcano in the solar system, actually the biggest volcano we've ever seen, Olympus Mons. It also has, for example, a canyon that is 3 times deeper than the Grand Canyon, if you've ever been there. And if you were to place it on Earth, it would go from LA all the way to New York. So basically from here to Montreal-ish. So it's really big. And Mars is actually 3 times smaller than Earth. So in terms of feature size, with respect to the planet size, it's kind of crazy. But Mars is also a single plate, what we call a single plate planet, meaning that it has no plate tectonics, but it has like one plate. So it's not separated. So we don't really understand, right? You know, on Earth, when we get earthquakes or mountain ranges, that's because of plate tectonics. So there's another sort of mechanism going on Mars that's causing, or that caused in the past, those effects. The other really strange thing about Mars is that it's actually evolved a lot more than our planet has. Mars, we know, used to have an atmosphere. It also used to have a magnetic field. Now on Earth, the reason why we have a magnetic field is because we have a liquid core that moves around, and it's a liquid metal core, so it creates that magnetic field. That magnetic field is actually the reason why we're alive. It protects us from so the sun is constantly emitting these energetic particles that essentially it would irradiate you. That's why astronauts can't stay in space for too long, for example, because they're damaging to cells. So, but the magnetic field actually protects us from those energetic particles and shields us, so they get trapped in the heliopods and then go toward the back of the Earth. And actually, the other thing is that magnetic field protects us from the solar wind. So space isn't completely empty. As it turns out, the sun creates this wind that is constantly hitting us. And if we didn't have that magnetic field protecting us from that wind, our atmosphere would actually get blown away. So Mars actually, we know, used to have a magnetic field because of the remnant magnetic field that we're seeing. And we also know that it used to have an atmosphere. So an interesting hypothesis that exists is that something happened to the core, maybe, right? If we take an Earth-centric model, what we could hypothesize is that something happened to the core where it went from being this liquid core that was producing a magnetic field to a solid core where the magnetic field went away, and then eventually that atmosphere would have gotten blown away. So that's what InSight is going to study, is it's going to study Mars and its interior, and mostly why it's so different from Earth and what happened to it. Now, one thing I like to point out sometimes is that actually this investigation could have been done at any rocky planet. We're actually interested in general about how rocky planets have evolved. So we could have gone to Venus or Mercury, and they each have this interesting history of evolution, right? Venus has a runaway greenhouse effect, for example. The reason why we're going to Mars, as it turns out, is just because it's the easiest planet to land on. It's the easiest one to get to. We've done it a few times. And so it was the easiest one to get funding to go to convince NASA that we could potentially do it. The results that we'll get from this mission, though, will affect rocky planets in general because it gives us an extra data point, right? We have the Earth, we have the Moon. By getting this extra data point, we can start to constrain models about planetary evolution. So that tells us not only about our solar system, but also about exoplanets, and it might give us clues as to what other exoplanets look like or how they might evolve and how to find Earth-like planets, right? And the essential question is, why is the Earth different? Okay, so how are we doing this? So this is a picture of the InSight lander. It is a lander. People look at this sometimes and they're like, oh, well, how does it move? It does not move. We don't want it to move because first it's easier, but the main reason is because the main instrument that we're using to study the interior of Mars is a seismometer. So we actually want something that's as quiet as possible. So a rover would not even be useful, even if we could have one. So that's actually the way seismometers is the way that we know about the Earth's interior. So what happens is whenever there's a seismic event, the waves will reflect and refract on the interior of the surface, and basically the time that it takes for those waves to get to you, as well as how much they're stretched or compressed, gives you an idea of what the interior structure looks like. So by looking at a number of those events, we can start to understand what the structure looks like, what it's made of, and how deep each layer is. So this SEIS instrument actually was donated by the French, so I get to go to Toulouse quite a lot to go to work with the French Space Agency. The other instrument we have is a heat flow and physical properties package, which is a handful, but it's HP Cube, which is a probe that was donated by the German, and it's going to hammer down the surface of Mars by 5 meters. So it's essentially a self-hammering nail that's about this big, and it has a worm drive on the inside that pushes it down, and it has a tether behind it. And what it does is it's actually measuring the physical properties of the ground. So it has a heater in the head of the nail, and that as it goes down, it will send out pulses of heat. And then the tether has little thermometers essentially inside it. So if you know what time that you're emitting the heat, you can also measure what the temperature gradient is along your tether, and that gives you an idea of grain density and composition. The other thing it's doing, people always ask me, well, why are you going to 5 meters? Why not 2 meters? Why not 10 meters, right? Why is 5 a good number? Well, what's important about 5 meters is once you get below about the 3.5 meter point, you no longer see any of the diurnal temperature changes or seasonal temperature changes. So day and night, winter or summer, you're no longer seeing that change that's happening on the surface. So all of the heat change that you're seeing at that point is anything that's cooling or emitting heat below the surface of Mars. So that might give you an idea of how the interior might still be cooling, or if there's any heat emitting particles below the surface. So that's the kind of the secondary investigation that's doing. And then with all those things, there's actually also the RISE antenna that you see labeled there. Those are a set of antennas where we're doing radio science. So using, if you've ever done this at home, I don't know if you ever boil eggs and then put them in your fridge and then don't remember which ones are boiled and which ones are raw, right? And I don't know, people do this apparently. So, so one experiment that you can do, or you might have done in school, is you can spin them, and they will spin differently because one has a solid core and one has a liquid core. So we have these antennas, and because we know where we'll land on Mars, and we know where the Earth is, and we know where Mars is, we can actually, with our radio signal, which moves at the speed of light, we can know, based on our distance, how much we're moving away from Earth or closer to Earth. So the only reason why that distance would be changing is because of the wobbling of Mars or because of the expanding and stretching of Mars. And that gives us an idea, the same as the egg experiment, about what the interior of Mars looks like. So that's a third investigation that's going on. We also have a robotic arm on the lander, which is the one cool moving part we have, because when we land, the 2 instruments are actually going to be on the lander. So we need to use a robotic arm to put them on the surface. And if you've ever been to the arcade and played that claw game, which I've never succeeded at, which is a little terrifying because it's basically that, but all the way on Mars. We practiced a lot. I swear it works. But so it does take us about 3 months to deploy all the instruments because of all the steps that we have to take. And we obviously can't do it in real time either because there's a 7-minute light time lag each way. So it's not like you can, move an arm on a joystick and it moves over there. We actually send commands in the morning. The lander executes them during the day, and then at night sends us back data, and then we code during the Martian night so that it can do its work during the day. So quick mission overview. We are currently in cruise, and then we'll be landing on Mars on November 26th at lunchtime. So it's the Monday after American Thanksgiving, known as Cyber Monday. So if you're shopping on Amazon or something, maybe you remember like, oh, I should go to the NASA website and look at what's happening. You might see me on console. But here's an idea of what's happening on entry, descent, and landing. And I'll show you this because as it turns out, the hardest part of going to Mars is landing on Mars. And so you can get an idea of a little bit about what that looks like. So we're currently in that cruise stage, and as we approach Mars, we'll get rid of our solar arrays, and then we have a heat shield, which is basically made of cork that heats up as you enter the atmosphere. The atmosphere is about 1% of the Earth's atmosphere, so enough that you still have to worry about it. So that slows us down, and basically what's happening is the cork is actually burning up, but we're sort of safely inside, shaking, but safely inside. Then we have a parachute that deploys, and it's a supersonic parachute, and that's when we're about 5 miles from the surface, so about 8 kilometers. And then we'll get rid of that back shell because we don't really need it. And actually we land straight with our lander, so the legs pop out. And at about 1 mile from the surface, the parachute is no longer useful because we can't slow down anymore. And we actually have thrusters on the lander that slow us down. So we have these thrusters that act the other way, and they slow us down so we can softly touch on the ground. And we have identified an area on Mars where we're landing, so we normally are able to land within 1 kilometer of the point that we've established. So yeah, that's what it's going to look like when we first land on Mars. And the whole descent stage takes about 7 to 10 minutes, not in real time, again, because of that light time delay. So all we get is the data back. One of the missions, that's not what I wanted, that I worked on before InSight is there's actually a companion mission to InSight called Marco, which is a pair of CubeSats. CubeSats are basically shoebox-sized spacecraft. They're about this big. They co-launched with InSight, and they're actually currently making their way to Mars. They're called Marco A and B, but we call them Eva and Wally kind of as, we're not allowed to officially call them that because Disney does not like people using their IP, so I'd not say that. But we call him even Wally because if you've ever seen the Wall-E movie, there's a scene where he gets ejected from the space station and uses the fire extinguisher to get back to, right, you've seen it. Well, as it turns out, the propellant that we use in Marco is actually fire extinguisher propellant, so that's why. It's super safe and it works, so yeah. So there's 2 little CubeSats. We're actually a tech demo mission. It's actually the first thing I ever worked on at JPL. And we basically are doing telecommunication relay for InSight as it's landing. InSight, we're kind of lucky at Mars because there's actually other orbiters around Mars already that will transmit that data. But we wanted to show that we can also do it with these small spacecraft. So if we're going to a target where there isn't an orbiter that can do that transmission, we're able to get that crucial data down using these really cheap throwaway spacecraft. It's also been really cool to show that these are some of the parts we ordered from Amazon, and some of them, they're like 3D printed metal that we made. So it's a very different paradigm from the multi-million-dollar missions that we work on, right? But they still work and they serve a different purpose. So as a newer engineer, that was a cool thing to work on. So yeah, I was a systems engineer on that. But in addition, I've sort of served a number of ranges on InSight, and I'll give you an overview, but again, I'll be here if you want to ask questions. But on the left, that's me in the clean room. So we have to, the same way as you use clean rooms, we use clean rooms to protect the spacecraft from not having bacteria or anything from us, or any dirt, because once it's on Mars, there's no AAA to come clean it. So we have to keep it as clean as possible. So a lot of my job there was to get all the instruments integrated on the lander and then test it. We actually test them in all sorts of different environments to make sure that they'll work when we get to the surface. I got to go to the launch, which was super cool. I actually got to climb up to where the rocket was, which was really awesome. So that's the closest I got to ever get to a rocket. And then and I work in operations. I'll be working in operations until we're done with the deployment phase. So we launched in May. We've actually been talking to our instruments for the past couple of months, making sure that they're all healthy, doing a bunch of diagnostic tests just to make sure that everything was okay. Our last test is next week, and then we turn off the instruments for the rest of the journey, and we'll talk to them again probably around November 28th, which is, I'll be very nervous to hear from them then. So yeah, that's kind of what I wanted to bring up, but if there's any questions or time for questions. I'm aware that it's like I'm the last stop between alcohol, so.
we do have time for questions. So please, students, ask questions. Anybody?
Did NASA get the idea from the Wall-E movie for the fire?
No. So as it turns out, the thruster that we're using is actually a coal gas thruster, and that's been a system that's been around for like 40 years, so definitely before Wall-E. But the reason why the joke came along is actually the thruster system that we can use can use different types of propellant, and one of them just happened to be our FA-236. So we just kind of one day put two and two together. Well, as it turns out, we were using a fire extinguisher as like our propellant tank in our test. So I think the idea, I remember when the idea happened, it was probably about 2 a.m. in the lab, and we were like, oh, Wall-E. Apparently these things stick, so be careful about what you nickname things.
Real loud, Adam.
In the past, I've seen them use kind of like a bouncy ball setup for landing on Mars for the very last stage.
Yeah.
What's the advantages and disadvantages of using thrusters versus
Bouncy balls, or even MSL landed with a sky crane, which is very different. So it depends on the mass of what you're landing. So with the bouncy ball system, that was for the MER rovers, which are much smaller rovers. They're more on the 100 kilogram category. I think they were about 80 kilogram. So that works. You can build bouncy balls that are big enough that can inflate enough to slow that down. InSight is more on the 300 kilogram size, so it's a little bit bigger. So because of that, we have to use thrusters. And InSight is actually a cousin, it's a replica of a previous mission called Phoenix. So it wasn't actually built by JPL, it was actually built by Lockheed Martin, so that's also that system, so we partner with them. So it's just that system works for that particular weight. And then the sky crane that you might have seen for Curiosity and we'll be using on other bigger rovers, Curiosity is more of the 1,000 kilogram size, so that's just what works for that weight. Yeah.
I assume that the surface needs to be really even for you to measure seismic activities and whatever other things you're measuring.
Yeah.
How do you ensure that the surface that you're landing on is even enough for you to measure all these things if your instrument can't move?
Yep, that's been the object of my nightmares. So there's two things. So first off, we actually have really, really high resolution images of Mars. So often people ask, how did you pick your landing site? Well, we picked the most boring landing site we could find, right? It's the flattest area. It's like what we call it's like the Midwest, the American Midwest of the Saskatchewan maybe, to put it in Canadian term, of Mars, right? It's super flat and there's nothing going on, which is absolutely what we want. So in that sense, we know that there isn't any big mountains or global gradients. And then we've actually practiced deploying our instruments so that they can be deployed, and they're built to survive up to 15-degree inclination, local inclination. So that helps too. And then our robotic arm actually has a range of about 1 meter. So if we land on a given area, there's going to be rocks, so we can actually try and dodge the rocks and put them in between. So between the images and the statistical models we have, we have a very high confidence that given our landing site, there will be an area that's appropriate for each of the instruments.
So on that note then, so how do you know that this bore that you're going to send in, that you'll be able to penetrate the
depth? So there's two things. So we have actually practiced in the lab. We have interns and undergraduates who will come in and see. This is the cool project you have at JPL. But yeah, so build buckets of sand, and so we've practiced digging into different surfaces. One of the interesting things is if it hits a rock, if it's not too sizable a rock, what it'll do is kind of, as it's digging, it's not guided, but as it's digging, it will automatically sort of start digging to the side and go down, because it doesn't need to be perfectly vertical. So that we know. One of the issues, though, is if we hit a big flat sheet of rock, we know that we can't go deeper than that. So I actually asked one of the scientists, well, how do you know what's below the surface? Because I've just always been told, well, there isn't any below 5 meters. So the way they do that is actually there's micrometeorites that impact Mars all the time, because it doesn't have the atmosphere that we have, right, to protect us from what you see as shooting stars. So when they impact, depending on the size of the meteorite, it causes different craters. So the craters have different depths, and the great thing about micrometeorites is when they hit, it causes this crater, and all of the stuff that's under the surface gets brought up. So they've seen that when you're looking at 4 to 5 meter craters, there's only small rocks, and the bigger sheets of rock happen in much deeper craters. That's how we know.
So how big is Insight?
So the deck itself is about this big.
Oh,
yeah.
it's small.
Oh yeah, it's really small. It's probably smaller than this table. And then the solar arrays are again this much, as much on each side. So it's fairly small for a spacecraft. Yeah.
Are there other cool NASA projects that you maybe want to pursue after this?
Yeah, well, so my dream, and I think you hinted at that, so I am really interested in ocean worlds because they have a potential for astrobiology, which is basically life outside of Earth, but in our solar system. So the best 2 targets for ocean worlds, for astrobiology in our solar system is Europa, which is, well, one of them is Europa, which is a moon of Jupiter that has a global ocean below its surface and then an icy shell. And its ocean is actually bigger than the Earth's ocean. And then my favorite moon is actually Enceladus, which is around Saturn. And again, it's an icy world, very similar to Europa with the icy shell. But the cool thing is at its south pole, it has these geysers that are spewing water. And that you could literally just sample. And we use instruments that you may be familiar with, like mass spectrometers, to look at. So we actually the idea would be to look at samples from either Europa or Enceladus and look at the composition and to see if there's biosignatures that could identify the building blocks for life. So there's actually a lot of chemists that work at JPL because of that interest in finding life. So my hope is to work on, we have an ongoing Europa mission, and then we've been proposing an Enceladus mission for a little while, so that would probably be where I would want to go after this.
So can we send Trump to check on one of those missions?
I don't, yeah, I won't comment on that.
I'm sorry.
I was just curious as to why they prioritize the Europa over Enceladus, because my thoughts are there are these massive geysers. If you just manage to hit near one, you could just sample the ground around and find something, whereas Europa, you'd have to drill through like a couple of kilometers of ice, wouldn't you?
Yeah, I agree. I really like Enceladus, and so that's what we've been telling them. Europa has just had other scientific interests in the community for a long time. Also, Enceladus had a lot of attention because of Cassini this whole time. So, and we have seen interesting things at Enceladus. So Europa has just been one of the missions that we also happen to have a congressman who really likes Europa. Europa has its own credits. I just happen to know more about Enceladus, so maybe I'm selling it more. One of the cool things about Europa that we found out recently from the Hubble Space Telescope, though, is that it might also have plumes. There's been 2 observations recently that have shown that, and actually old data so we used to have a spacecraft around Jupiter that did flybys of Europa, and so it did something like 20 flybys, and one of them just happened to be really odd. And so you'll discover this if you continue doing research. PhD students do all the research and discover everything, and then professors just get credit. But it's true. But what happened is there's this professor that remembered this flyby and told her PhD student, go investigate this. And they investigated that flyby and the timing of when it would have happened, and discovered from 20-year-old data that actually they had observed, instead of being an anomalous flyby, they had actually observed the plume directly. So Europa might actually have plumes. That guy did get his PhD super fast too for that discovery. But we think that they might be seasonal. It's not clear exactly. Maybe the density is different, and that's why we haven't seen it before. So we are flying the right instruments to detect molecules from those plumes if they do exist. Yeah.
So how long do you think it will take to get all the data?
To what?
To get all the data.
From InSight? So let's see. So the full mission, the nominal mission is a 2-year mission, so it's 1 Martian year, mostly because of that probe, because we want to see the variation. For the seismometer, I mean, if the big one hits the day we land, well, not the day that the instrument's on the surface, then that's a huge plus, right? The interesting thing is that there's models for what we think the seismic activity looks like, but we're not sure, right? They're all based on hypotheses. So there's no plate tectonics, so we think there's Mars quakes, but we don't quite know for sure how they occur. It would probably be because of magma below the surface causing it. And then otherwise, if there is no tectonic, activity, we would still get those waves from micrometeorite impacts, so then statistically we'd have to see how often they occur. But really after 2 or 3 Marsquakes, most of our questions can be answered. So, but in terms of like cool things for you guys to see, we'll probably be transferring, like the first thing that we do almost is take a selfie of the lander. Not because we're millennials, but because we want to make sure that everything is on the deck, still in the right place. So those pictures will be released like within 1 or 2 weeks. So, so then it just depends on how long the peer review process takes.
What will you be controlling during the landing?
I actually work on the instruments. So during the landing itself, I will probably just be watching and drinking wine, trying not to freak out. So I will not be on lab. But so during landing, we don't actually control anything. And that's kind of the scary thing as an engineer, is everything is done ahead of time. So we target the planet. At this point, we are so well targeted that we already know where we would land if we did nothing. And everything is pre-sequenced and tested. So when you see people on console on landing day, whenever you see those cameras, those people aren't doing anything. They're literally sitting there looking at their screen because there is nothing you can do. So that's why it's a little nerve-wracking. And all we do in real time is reconstruction of what's happening. But yeah, so before that we target where we're going and target the timing and things like that.
So how do you account for environmental changes, like unexpected changes?
When we land, we don't. So for example, one of the scary things that's happening recently is there was a really big storm on Mars, for example, but we are over-designed to a point. So what we do is we look at historical data of the atmosphere, and we have models for what the atmosphere looks like, and then we design beyond that so that we can deal up to like the 3 sigma case. But we can't in real time change anything. The thrusters are what they are, but in real time, what the spacecraft does do, right, is it has some intelligence. So it knows, for example, its velocity. So if there's more or less atmosphere that day, based on that, it will thrust for shorter or longer. So it does have some idea of how far it is from the surface and how fast it's going. So, but that we don't, the spacecraft does in real time, we don't.